Hello, everybody. I think we should start. So welcome. I'm Prabhat Jha. This is my inaugural opening of an inaugural lecture. And I'm delighted that we're going to hear from Goylette Chami today. So I think many of you know Goylette from just her remarkable and visible presence. She's just a force of intellectual energy and enthusiasm about global health. So Goylette joined in 2019 and has really grown a remarkable program, particularly on schistosomiasis, on which she'll tell you more, including a Oxford, Uganda collaborative program. She's worked very closely with Rory Collins and Martin Landry, both of whom were not able to attend today, but send their regrets of being unable to. And I hope we will learn a lot. I'm looking forward to learning from her own experience. Just before this, we were talking about the global burden of disease numbers, which estimate only 6,000 deaths from schistosomiasis, which Goylette thinks is quite an underestimate. But we'll hear more. So with no further ado, let me introduce Professor Goylette Chami.
Brilliant. Thank you, Prabhat. I didn't want to give a guess on what the GBD estimates to be mortality related to schisto because I'm not comfortable giving uninformed guesses. So I'm really excited to be here today to talk to you about my favorite pathogen. I've been working on schistosomiasis, and I want to share a bit about what's motivated me and how I think about this problem. I say my favorite pathogen, which I think comes naturally to parasitologists, but my colleague Sarah Lewington looked at me like I was crazy. And she was saying, well, people that study smoking don't hold up their favorite brand of cigarettes and say, this is my favorite killer brand of cigarettes. So maybe it's something strange about infectious disease researchers that we are fascinated by the pathogens we study. And I hope I convince you today why schistosomiasis and the schistosomes that cause it are really interesting. It's a disease of poverty. It affects over 250 million people in low-income countries. Over 700 million people are at risk of infection. It's mostly in rural sub-Saharan Africa. It's really beautifully complex in its life cycle in that it's transmitted from humans openly defecating or urinating in freshwater sources. There has to be a competent snail. The snail has to further the parasite's life cycle, has to go and find another human, has to survive through the human host, and then it has to go on through that process again. It has really complex social factors related to it in the administration of treatment, the inequalities in the access to healthcare. So I pretty much think that you can study anything you want by studying schistosomiasis. And this is kind of the approach that I've taken in building a group around it. It's both beneficial and harmful. There's someone in this room, David Dunn, who has worked on it as a misguided allergies kind of being a misguided anti-parasite response. So if you're ever going to find anything that might cure or help allergies, you look into something like schistosomiasis or other helminths. Of course, it's also deadly. It can cause severe liver, high blood pressure, portal hypertension, which is what I've been focusing on. And there's a lot of room for discovery. Actually, surprisingly little is known about the epidemiology of this neglected tropical disease. And that's because it's thought of as something that's quite easy to target rather than a chronic infection that results in lifelong disability. So there is a lot of room to answer actually relatively simple questions about how this parasite behaves, who is it affecting, what disease does it cause, and what are the timelines for that. And it's also, in my view, and this is kind of more of my future vision, is that it's an ideal platform for understanding questions of global health importance and discovering antifibrotics. Schistosomiasis causes fibrosis in really quite diverse forms, quite rapidly. So it's really interesting if you're ever going to find a population that's heterogeneous enough and progresses quick enough to discover new therapeutics. But of course, I didn't know what schistosomiasis was when I started or when I went to undergrad. So how did I even get here? I was really interested in poverty. I mean, at that stage, I couldn't even pronounce schistosomiasis. I came from, people always ask me, your name is spelled weird. I always have to give you an explanation. Well, here's the explanation in one go. My parents are Syrian. I wasn't born in Syria. I was born in Budapest, Hungary, in the last years of communism, where my parents were sent over to work for the communist government. But I was raised in the US, so my name ends up being a product of mistakes. I actually went to Washington Community High School that got abbreviated WACO High. Maybe this is like an indication of my career of starting off. After that, I didn't know what I was doing. I was a first generation university student. I went to the best university that was nearby, and that was the University of Illinois at Urbana-Champaign. I actually studied economics and political science, and it sounds really strange. Now I'm an epidemiologist. I focus on clinical problems, but I can look at this room. I can think of the people online. A lot of us now are coming from different career paths. I don't think you start off in one thing and you stay in that now. So this is pretty normal. But I did that because I thought I was going to become a lawyer. So I was on this path to do pre-law, go and do a JD, maybe a PhD JD within the States. So I went abroad and I had a study abroad in Corvinus University in Budapest, which was actually my first time visiting as an adult. It was formerly known as the Karl Marx University. I came back, I founded this organization within my undergrad. I was very keen to do stuff, anything related to poverty. I gave it a terrible name called the Global Entrepreneurial Development Initiative, not realizing that it's abbreviated JEDIs. I have never seen any movies associated to that. It's actually now been, and for a long time, it's been acquired by Entrepreneurs Without Borders. It was a microfinancing organization, and we went to war-torn areas of Croatia and partnered with the University of Zagreb and the University of Osijek. So that was really quite eye-opening for me. I went on, I had another semester where I actually interned at a law firm in Syria. Again, I was still thinking about this path to a JD. But then I had these really, really important mentors. So I had Paul Magelli, Peter Narduli, and Joe Hinchliffe. And most of these guys, this is an economist, a lawyer, an economist, and a political scientist. And they sat down and they said, well, you're interested in studying poverty. How do you think you're going to make an impact? Do you want to be in a courtroom? And I said, well, no. And they're like, well, you need to go to grad school. I think you're not thinking of the right thing. So thank goodness they saved me. Had a failed Rhodes interview, few, because then I went to the other place, Cambridge. Not few because Oxford is terrible. Clearly I'm here and I love it. But few because it was the wrong degree for me. It was staying in the paths of economics and maths, which I don't think would have kind of broadened my interest. So luckily I got this Jack Kent Cooke scholarship, which was 6 years. And they said, you can study whatever you want, go wherever you want. You can change between your master's and PhD. It was the most beautiful thing I've ever had, complete freedom. And I came over to Cambridge. So at that point, I really started getting into infectious diseases, but I started off focusing on still kind of an economic viewpoint and using game theory to understand how do you coordinate behaviors to prevent the spread of certain pathogens. I was looking at waterborne diseases from bacterial pathogens and building these really simple coordination games. Like essentially, if everyone doesn't do the same thing and you have one person contaminating the water, there's no point in anyone being safe. Yeah, that probably wasn't a good conclusion, but that's what I came to at that time. And during that, I was still on this kind of, let's go places. I had never been to a low-income country. We organized a group of students from my MPhil, and we partnered with the UN World Food Program, where we were interviewing former child soldiers from the Lord's Resistance Army and thinking about their reintegration into these communities. So that was a lot of fun. And that actually was my first trip to Uganda, which my team online will be a bit surprised. Actually, that's how I started off in 2009. We visited the parliament. We met with the minister of microfinance there. We partnered with an AIDS organization to understand what was going on within the communities and conducted focus groups. It was really eye-opening, and I wasn't going to go back from that. I absolutely loved it. But things came on hold. Life is obviously not linear. I went back to the States. I stopped all my studies after I finished my MPhil. My mom was terminally ill with cervical cancer, and I took care of her. My stepdad, who was my kind of dad who had raised me, had already passed away during my undergrad. So this was a really tough period for me and kind of a turning point of deciding what I was going to do with my life. But actually, the good from the bad, I because we were going around trying to find new clinical trials for her advanced form of cervical cancer. That kind of exposed me to those methods and appreciated actually the robustness of clinical trials. It extended her prognosis by quite a long time. So that was important. I read this, which I feel almost every medic probably will tell you that's interested in tropical diseases. They've started off with Mountains Beyond Mountains. It's about Paul Farmer's life, a professor who has passed away from Harvard, who had found partners in health. It really inspired me actually, and it kept me going. So I knew I had something to go back to. I had an offer from Cambridge, and importantly, I had the funding that was letting me go back, and I had really supportive supervisors. So I went back, tried to have a little bit of fun in the meantime, did a bit of rowing, made Oxford a little bit annoying, played for the university and played basketball. So that was quite fun. I wasn't around a lot because I did a lot of field work, but it gave me a good balance. So in Cambridge, I had these fantastic supervisors. These guys are all either online or in person today, and I cannot thank them enough, my supervisors and my mentors. So Professor David Dunn was thinking, what am I going to do with this person? He's an immunologist, hosted me in his lab for immunoepidemiology, but was great and was tolerant, and I really tried to put in the work to understand the biology. Andreas gave me the quantitative perspective so I can continue with that type of work in econometrics and behavioral economics. So econometrics is not so different than statistics and epidemiology, just thinking about those disciplines. So I really wanted to keep my strong quantitative background. And then David decided, well, maybe I was too much. I was his last PhD student. So he moved on. I'm just joking, but he was in charge of Cambridge, Africa. He had founded this program, which is phenomenal. It's what AFOX is based off. Lots of African capacity building programs are based off. And just by kind of being able to observe and be around those programs and its events, I could see what it was like to have meaningful African partnerships that were more equal. So that really influenced me. David, at the time, was the head of the Liverpool School of Tropical Medicine, and we met fortuitously because I had emailed him to ask for a paper thinking this famous professor's never going to respond to me. And he did. And he was so generous with his time. And he's probably the only one for some odd reason who had blind faith in me and supported me meeting people and starting off with my career. But he introduced me to the wider neglected tropical disease network. And also Alan Fennick here, who was the founder of the Schistosomiasis Control Initiative that really gave me deep insight into how schistosomiasis treatment was distributed and the way to approach that. So I had this great academic balance, and I was really, really, really well embedded in the policy world and the WHO world and the reality of what it's like to give treatments on the ground. So I was excited. I was going to work on the largest treatment program in the world, which was mass drug administration. So for those of you not familiar, this is actually meant to treat a number of neglected tropical diseases, not just schistosomiasis. You don't diagnose people. You go to the communities. It's safe to administer, often as a single dose. And it doesn't matter if you're infected or not. You go door to door if you have community-based distribution and treat everyone, or you distribute through schools, but you're relying on these lay health workers, kind of volunteers, to reach as many people as possible. Over 1 billion people at that time. Now there's international funding cuts, so maybe not so much, were targeted and treated annually. And I really wanted to work on these programs and to understand them a bit more. So the idea was I wanted to work on this new field of developing network-based methods to evaluate these mass treatment programs. I wanted to know what determines the reach of these programs. Are we reaching the right people? How do we better select the medicine distributors that are involved? And how does information spread about mass treatment so that you get people to take it up? But I had to have somewhere to do the field work. And this is, I mean, again, this is a path of really fortuitous meetings. So then I met Lorenzo Savioli, also through Alan. He was the founder of the WHO Division of Neglected Tropical Diseases. Maybe this counts as my first WHO-sponsored trip. Well, he was interested in the ideas that I had and said, why don't you go out to Zanzibar? Why don't you learn about what's going on and tag along? Professor David Rawlinson, he got the task of babysitting me. Didn't know I was coming. This is a photo from us being on like a 4 or 6-seater propeller plane where I sat in the co-pilot seat. I mean, I don't know how old. I must have been about 23 at the time. I was giggling in the front seat. I'd never been on a small plane like this, but it was great because David showed me around and I got to see my first kind of primary schools where people are getting treated, where the transmission sites were. I got to talk to people that had so many years of experience. And I was there at the time where they were launching this huge, huge campaign because the islands of Zanzibar, well, if you have an island, maybe you could eliminate a pathogen. So Gates had put in tons and tons of money in an attempt to have the first elimination of schistosomiasis within a sub-Saharan African country. And I got to learn how that trial was set up, all the different interventions they were doing, go to the different campaigns. Unfortunately, it didn't eliminate schistosomiasis, which we're still here today. Quite difficult, but was really quite exciting. And then in that, I was put forward to try to pitch my ideas to people at the WHO and the local Ministry of Health. That was kind of by Lorenzo. It gave me a chance to kind of, I guess, I don't know, be on the iron or whatever the phrase is, where you have to convince people that your ideas are good enough and then go forward with them. So that was definitely an experience. And it also exposed me to the realities of fieldwork. I remember sitting in a room and these two postdocs debating about who was going to take, for better word of lack of profanity, the poo. And I didn't know what they were talking about. They were arguing back and forth. They were genuinely debating who was going to take a suitcase full of poo samples at that time on the plane with them and home. And I was like, okay, what am I getting myself into? I came from economics and a maths background, but it didn't scare me off. It was great. So I didn't end up doing my work in Zanzibar. They had a trial there. So get off the island, go somewhere else where you're not going to contaminate the trial. So David Dunn and also Alan Fennick had long-term connections to Uganda. And this is where I met really my lifelong friend, my mentor, and collaborator, Dr. Narcis Kabatrani, who also introduced me to Dr. Drita Tukahebwa, who is the head of the Division of Neglected Tropical Diseases at that time. Narcis was not as excited to meet me as I was to meet him. I knew that he was a really established researcher. He's absolutely brilliant. But when I showed up, he was thoroughly disappointed because he thought Professor David Dunn was coming. And it was me on my own, about what, maybe 23, 24. And he wrote to David and said, "You have sent me a baby. What do I do with her?" Yes, I hadn't done any field work yet. I wasn't sure what I was doing. And he tested me. He tried to put me through the ringer. He would take me to the worst places to stay, where there were holes in the bed net. We'd pull the blanket up and you'd be bit. Where it was in rougher condition than it even needed to be at that time. But I survived. And he's quite proud of that. So he thinks I'm now a little bit tougher of a cookie than he thinks. And this had lasted. So where are we going to get the money to do this project of setting up networks and evaluating mass drug administration? Maybe this is not a nice saying. I wasn't sure, but I did Google it, and it does just say you put your money where you have faith or you believe in someone. And David Dunn took a punt on me. Well, he was my supervisor. He was like, well, why not? Gave me a bunch of welcome money. Alan took a lot more convincing, lots and lots of meetings and proposals and official pitches. And Narcis takes the credit for convincing Alan eventually. But Alan, against a lot of people's votes, let's say, in the organization, also put in a ton of money. And then we have our collaborator, Erwin, who was a collaborator previously with Andreas, my other supervisor, who just thought, this is a cool idea. Cool, I'm on board, I'm in. I don't know what any of these people are thinking right now. My students are fantastic, but I'm not sure I'd give you a couple $100,000 to go for it. And they did. You only get one shot in field work. Thankfully, it did kind of work out in the end. So I set up during my PhD this Uganda-Cambridge collaboration on mass drug administration. And this was the largest study ever done on kind of social networks and health advice networks in sub-Saharan Africa. We went to 17 villages and did a complete census of network connections between everyone within those villages. So we surveyed over 16,000 people. We were based here around Lake Victoria in eastern Uganda in Mayugi district. We collected lots of diverse information, all the GPS points of every home to find out where the drugs were actually going and who they were reaching. We did follow-up surveys after the mass treatment campaign to see whether people were refusing drugs, who took up the drugs. we did lots of infrastructure surveys, school diaries, and then the clinical measurements at this point were rather simple. We were focusing on infection and anemia. It was tough. This was in the era before large electronic kind of surveys were commonly used. So we bought 100 tablets, worked with them. We're one of the probably largest or first users of ODK at that time, Open Data Kit, programmed our own surveys. And then in the craziness, I went around with two of my kind of close collaborators at that time, Benjamin here and Juma, who's the district health officer, and we were giving literacy tests to everyone in the village to try to recruit local villagers to be part of the study so that we had trust within the village so that you could go and ask them about their connections and also for them to be part of it. The only requirement was they had to have kind of seen a phone and used a phone, not a smartphone, a button phone. And then we would work with that. But who was I to think I'm going to interview people in Lusoga, a language I don't speak, and select them? But it did work out. We trained actually almost 200 people with detailed like open data kit protocols, all these electronic surveys. It was a huge, huge success. Of course, fieldwork was tough. This was my first kind of real dive into it. When I was looking for photos, I found a document that I titled Fieldwork Surviving, which I was a little bit surprised to help me get through the field. We were working at times by candlelight. We didn't have servers where we could send things to electronically. I was sitting there with our 100 tablets, plugging each one in one by one every single evening, battling the rats in my room, which were probably my own problem because I was a bit of an idiot and had fruit in my room. So get these giant rats that would run through. It was tough, but it was really, really rewarding. And we got to work with all the community leaders, the village health team, and had a fantastic team. So what did we learn out of that? Well, sadly at that time, when we did this door-to-door evaluation of the mass treatment programs, we saw that actually they were missing individuals that were most in need. It was believed before we had gone in, because of some small-scale studies that were done by anthropologists, that people were actually actively refusing treatment. But what we found was that barely anybody refused treatment. It was an issue of not reaching them, of not them being offered the medicines in the first place. In the villages we went in, overall, there were greater than 50% of individuals that were missed during the campaign, and most of them didn't know about the medicine availability, despite the medicines being in the village at the time with the drug distributors. When we looked at the models of who actually was missed, they tended to be marginalized households, so low socioeconomic status, no home latrine, poor water sanitation and hygiene behaviors, and belonging to minority tribes and minority religions. And then unfortunately, also some heavily infected with schisto were missed. But all of these, importantly, again, were correlated to the medicine delivery and not to the compliance. So now it became an operational issue. It wasn't the fault of the participants for not getting the drugs. We also, of course, the networks-based side of things, where that social networks matter. Okay, surprise, surprise. But we find that the missed households they matched the marginalization characteristics, but another characteristic of marginalization was simply being on the periphery of your social network within your village and thinking, even though these are small rural village networks, information is contained, it might not spread so easily. The people who actually had the most friendship connections were the most likely to get treated. But there's wide variation by village. So some villages might reach about 23% of the people, going up to 94%. And it was quite interesting because there's quite a few papers about information diffusion and how that spreads. And if you think about networks, you might think you only need to have one contact for information to spread or for you to take up an intervention. But what we found was actually it depends on the kind of the density of your contacts and how closely knit your network is around you for being reached during mass drug administration or taking up the intervention. So this kind of proposed a new hypothesis around complex diffusion, and you might need reinforcement. So you can't just hear about from one person about the campaign, but you need to hear about from several people about the campaign. And we saw that the difference in drug distributors that also had these closely knit networks also meant there's a difference in the percentage of households reached by about 10%. But what was most important was those structures, those closely knit structures, could predict how quickly you could reach people within the community. At the time, these programs were set up so that you'd have a week for a particular drug, another week for another drug. We actually monitored them for quite a bit longer, for 31 days, and looked each of these plots as a different village, how long it took to meet a particular threshold that was targeted either locally or that was comparable across the villages. If we take a very low threshold, a low bar of 50%, the drug distributors that had these very closely knit connections, they actually could reach 50% of the households within their villages in half the amount of time. It would take them 10 days versus 21 days. So this was actually quite exciting, and it kind of went against the knowledge in network science at the time that you just needed more contacts and didn't matter about your density or clustering of connections. So the overall implications of this were that, well, actually, the low treatment rates are due to poor medicine delivery, not compliance issue. We actually end up missing people systematically if we can profile them. That was hugely important to how we modeled goals for how long do you need to do mass drug administration, because it was assumed, let's try to target, let's say 75% of individuals, but the 25% that are missed are random. But if you don't miss them at random, you can still undermine controlling whatever you're targeting, whether it's some sort of transmission target, which is usually not the case, or some sort of prevalence target of infection or future morbidity control. So that was really important from this. And that blanket treatment could potentially be inequitable. So this ended up getting invited for a piece in Lancet Infectious Diseases and kind of went on from there to inform a number of policies. I went on after that to actually try to hone in more on the clinical side of things and thinking about interventions, and did a junior research fellowship. So I didn't do a postdoc. A junior research fellowship, it's independent, but I didn't have a group. So it was quite nice. I still had the freedom. I was still in Cambridge. And that was at the beautiful King's College, Cambridge. This was great for me because being a fellow in a college meant that I could sit next to a medievalist, a lawyer, whoever at lunch, and just, if they would listen to me about worms, talk about my ideas, and also hear from their methods. I see people laughing here because you have heard way too much about my worms, I'm sure, at lunch when you don't want to hear about it, but you're going to hear about it if you're next to me. So that was great. I got to tell lots of people about worms, and I tried to tell more people across the university by hosting these global health seminars that actually gained so much attention. The Lancet Infectious Diseases opted someone to come and write a few pieces on it, so that was fantastic. I was holding a couple other joint positions. I wasn't there for very long. I actually left that fellowship early to come to Oxford, but during that time we expanded our work in Uganda to more villages, and we did a bit of work to figure out, well, okay, if we are missing people systematically, if we don't have the coverage rates that we anticipate, there should potentially be residual disease that we'd expect related to schistosomiasis. A few things we thought about, though, before that was thinking about how can we take some network methods and make them into very simple interventions. Even though we had focused on clustering, there was still a worry about, in some communities and other places, of rumors spreading and potentially stopping the campaigns. So we worked on how do you essentially destroy a network to stop something from spreading, and looked at these friendship and social networks at that point, and just exploited a very simple mathematical principle. If you have a network where you have a few nodes, people connected to many people, they have to then be connected to people that have few connections. So if you were to randomly sample, let's say a household, and you would look within their connections, you're most likely to find a hub, because your first sampling is that you're going to get something with probably few connections, because that's most of the people in the network. Your second step is that they're likely to be connected to a hub, because the hubs have to be connected to the lowly connected households, because they have so many connections, they can't only be connected to themselves. So we're trying to exploit the simple mathematical principle to show that actually, even when you seed interventions and think about who you select, or you think about how to fragment a network, you can choose people better than who we tend to choose in the mass drug administration campaigns, which are, unfortunately, you choose the village health team, you choose the community leaders because you think they're very well embedded in the network. But we can almost always find somebody who's more embedded by using this principle. That was kind of the short and sweet of that. we went on then when we were looking at residual morbidity, the short story, which has inspired the rest of my career at Oxford and everything that I'm going to focus on is that there was residual morbidity. And it's quite complex. It has so many different causes, but the most complex cases, they weren't even being seen in the government facilities. They were going to private care. And we developed methods for kind of soliciting medical histories and thinking about how you process those layman reports of medical histories into international disease ontologies that are quite distinct. So that kind of built a foundational basis. Now, this was all exciting. It was getting kind of some research and policy attention, but then also some media attention, some that went better than others. This is, I was involved with the Cambridge Science Festival where they asked if I would be a featured speaker. I didn't have like great experience with media and whatnot, so I thought it would be wonderful to hold a giant jar of ascaris or roundworms, which Shona kindly lended to me from Cambridge. And instead, it turned out to be a very bad public health campaign of all the diseases you can get from different types of worms. Yeah, so this talk was targeted towards young girls because it was sponsored by a school within Cambridge to encourage girls to go into STEM. So I prepared the whole talk around it with my nicely advertised video to tell them about all the great work we had done. Sadly, none of those kids came. One young boy came. I think he was forced by his mother. I learned very quickly that when you do science festivals, the age demographic is much older. So I tried to insert a Justin Bieber joke that fell very badly, but at least it was getting some attention, right? Well, the Ministry of Health thought it was interesting, and there was a big workshop, and I was involved with that and contributing to what they were thinking should be new kind of targets and guidelines. But then, of course, I thought I never have my work associated with the movie Clueless. So the whole intervention of how do you select the popular people somehow turned into or not popular people, how you should select people in networks would be, should we treat and educate popular people first? And then there's a long story about how this work should be translated into the movie Clueless. So that was strange to say the least. So I left the JRF early and came to the other place here where I've been ever since. Tried to balance, I wasn't going to be playing basketball anymore. I can't keep up with the undergrads and who not. So I picked up running. So if I'm not here, I'm most often here, probably the last one. But if I'm not, you can see me volunteering at Park Run or running with Headington Road runners in the mud, or ideally in the mountains. So a lot of you, if I'm not talking about worms, I might be talking about running. Sorry. But I came to Oxford in 2019, like Prabhat said, on a Robertson Fellowship or grant. Actually invited first to give a talk by Rory Collins, who then went to schedule lots of meetings for me that I had no idea what it was about. Lots of half-hour meetings with different professors within the department, not realizing that maybe that was a potential mini interview. I was completely naive, but it was great. And then Martin Landry at the time was the head of the Big Data Institute. So Rory was the head of department and Martin the head of the Big Data Institute. This was brilliant. So I had people in my PhD that had this blind faith in me, gave me loads of money. I had this one shot. I did field work. It seemed to work out. Then I came here and they're like, okay, go for it again. Here's £1 million of a discretionary fund. What can you do with it? So I was super happy. Then there's lockdown, of course. So what could I think of other than to aim quite big and to try to set up a cohort? I'm not going to sit here. I have this time. I'm stuck. So I was focused on thinking about how can we build a prospective community-based cohort where we track individuals over time in Uganda to understand the disease progression. And that's what I worked on when I first came, in hopes that we can design better treatment in any, because there aren't really any guidelines or in any primary healthcare facilities, disease management strategies for schisto. But I do want to take just a second to mention the research environment here, because I was really, really fortunate. I think I had mentorship from Rory and Martin where they both, there was no pressure to produce a ton of publications, to bring in tons of grants. It wasn't quantity based. I felt really fortunate for that. They understood that kind of big questions take time, especially if you're looking at disease progression where you need to follow people for years before the bigger papers come out. Advised me to say no to distracting opportunities because things were getting exciting and I was getting invited to be involved in a lot of things. But the best thing I did was say no to everything and just stay with schistosomiasis and nothing else. So many will know, even now, I say no to everything unless you can link it to schistosomiasis, which we can link many things. and to choose the right people to build my group. So I definitely was a small fish here. This department's incredibly impressive with people's methodology, so I was quite inspired and I've learned a lot since I've been here. I have new collaborations across methods, across the division, across topics. The students are great. They're very quantitative. The epidemiology training is great. So really, I felt like I had the environment to kind of run, and I'm really happy I've done that. So I've mentioned that I was stuck within this kind of year or so or two of lockdown. So we worked on setting up the research backbone of the cohort that is now SchistoTrack, which I'll tell you a bit more about it. I started off with only one research assistant, a former MSc student of mine, and we would sit there programming surveys kind of day in and day out. I worked on setting up kind of all the survey designs that we were going to use, but a lot of time, and I think people will know this when you're doing these huge collaborations, was spent on setting up like infrastructure for computing, which I'm really grateful for the BMRC here. We were a guinea pig of setting up kind of private clusters and working with that. Years of data governance and contracts, research contracts. But at least I wasn't going anywhere for a bit. Worked on building the Oxford group. I'm so proud of this group. I have to admit, I started off wrong in thinking about how you recruit people because I wanted someone who was going to understand in depth the biology and do super in-depth quantitative methods, essentially do it all. And I don't think that is the right way to build a group. Instead, I focused on different methods backgrounds. And I was convinced I will teach anyone like the schistobiology or the schistoepidemiology, as long as we can bring different perspectives and expertise within the group. And that's what we have right now. A few people aren't pictured, but we're quite a diverse group in Oxford with epidemiologists, mathematicians, all the way to bioethicists. And the idea is that we're trying to capture the whole spectrum of problems in the process of understanding schistosomiasis from the discovery of the disease progression to the patient care itself, which I think works out quite nicely. And then when I was looking back, because I keep track of everyone who's gone through the group, I was actually quite amazed that exactly 50 people have been through my group in Oxford. That does include some master's students and interns, but still, I was really quite happy to have mentored these individuals. People who have gone on, they've gone on to stay in infectious diseases and work on global health when they haven't had any introduction to that before joining the group, or a really good fellowship. So again, a lot of the work I'll show you later will be from this group. The second group, which is really the same group, is my Ugandan group. They are probably even more closely knit than my Oxford group, who have been working with us for years. I can't introduce all of them and their specialties, but they call themselves the SchistoTrack family. That's great. I'm all for it. And we have more than a village as part of that team. We have specialists that are looking at the snails, individuals that go and get the medical histories and all the socioeconomic information, a variety of technicians with different specialties, nurses that help with the care and the abdominal palpations, and sonographers who are looking at our main outcomes of liver fibrosis. So all of this led to SchistoTrack, and it was quite a lot of work to put it together. And the aim of SchistoTrack is to discover the risk factors of liver fibrosis related to schisto and interacting with other causes. We're set up in 3 really diverse districts of Uganda. So Pakwach here by the border of the DRC, Bulisa on Lake Albert, and then Mayugi, where I was before, in eastern Uganda. These populations are really heterogeneous in terms of their ethnicity, the prevalence of their disease outcomes, and their risk factors. So it's actually a fantastic place to study liver fibrosis, but also hopefully to make an impact by improving the care of these individuals. We're in 52 villages with over 2,100 households. We've surveyed over 8,000 people. And clinically each year, we follow up over 4,300 individuals aged 5 years and older. So it's really a young cohort. I'm hoping this will go on for quite some time, rather than just starting with individuals that already have advanced disease. And the baseline of this cohort was in 2022. The outcomes we're looking at are quite diverse. The motivation behind this is that schistosomiasis, if I were to explain it to you, is not a single disease. It's really complex. It involves many conditions that have complex causes as well. So a couple of key things we're looking at would be co-infection interactions to those complex conditions. So we have malaria, hepatitis B, and HIV in the cohort, which have proved really interesting. And then we have lots and lots of liver and spleen pathologies, which are our main focus. And then some non-communicable diseases to look at concurrent outcomes, not necessarily interacting, but looking at multimorbidity in these populations that are living longer, that have a burden of both infectious and non-communicable disease. So I just wanted to share a few photos from the field. Both Rory and Richard Pito have repeatedly said this to me. A large study is not a small study done larger. This is a ton of work. Everything we do is at the point of care. If we need a bed, we make a bed that's foldable that we can take and build ourselves. We bring out all the ultrasounds. We set up in a primary school or a local health center. There's no record linkage. There's quite little health infrastructure. We coordinate, we bring out doctors to the village. So really we come out with these massive, massive buses with everything we need, with incentives for participants. It's a huge, phenomenal effort by both the Oxford and Ugandan teams. We set up these clinical stations where we have individuals come by and they're dropping off their samples. We do a number of tests. We look at anthropometry, at that time, blood pressure, height, weight, other things of that sort. We look for spleen and liver enlargement and other kind of clinical characteristics. We do really detailed ultrasounds and transient elastography. We feed back and coordinate all of these results so that they go to a treatment station and they can get treated or referred to on the spot. When we had HIV screening in this cohort, we even had immediate antiretroviral therapy enrollment and even sending people home with that, coordinating with the HIV counselors from the districts themselves. But if people need further care, we take them there. We call the doctors. We try to make sure that they're not just left and it's a referral to nowhere. But what can go wrong? So great, we have this cohort going. We can do all these interesting analyses. Each year of the cohort, something dramatic has happened. So we've had COVID-19 lockdowns to start, an Ebola outbreak, DRC rebels kidnapping young schoolgirls, an Mpox outbreak, and Ebola again. So this has made it a little challenging, but we've persevered even through kind of daily challenges. We commute through a safari kind of national park to go to one of our districts. So sometimes we have an invasion of hippos and other things and crocodiles. Actually, we don't have a problem so much with recruitment. Too many people want to be part of the study. So you get a problem of impersonation where you're trying to understand who is who. So we spend tons of time making sure the right person is there. But it all does work out. And we've learned quite a lot and have persisted. I wanted to start with one non-clinical result, because setting up a cohort, applying for grants, getting interviewed, designing the study protocol, participant dropout is always thought of as they've dropped out once, they're gone, you never see them again, a true dropout event. And that just doesn't happen. When you work within these really rural communities and they're quite mobile, they will rejoin the study. And this made a number of challenges for us in thinking about when you're setting up the initial sample size calculations, when you report to ethics committees, we had to convince people that people would come back and incorporate that in our study design. So in SchistoTrack, we find that actually only 7% of individuals are true dropouts. The rest of them will drop out at some point, come back, have different sequences, but they will rejoin. And that 7% is even high because it includes deaths. So now to move on to the main outcome, which very briefly, but I'm very excited about, is periportal fibrosis. This is the form of liver fibrosis we focus on in the cohort. So with schisto, you have the adult flukes living within your circulation. The female flukes are producing eggs. They're competing against the flow of your bloodstream. They'll get swept back within the liver and the spleen and induce a granulomatous response that, if it's uncontrolled, could lead to fibrosis or scarring, for those not familiar. Now with schisto, in the portal vasculature or in the liver vessels, it's really phenomenal because it can result in restructuring of the vasculature of the liver. The liver is an incredibly resilient organ. And we try to look at these changes on imaging and track that to look at the disease progression. So it's something we've been focusing on within the cohort. We are pushing for more imaging and better diagnostics because actually the way that severe outcomes were thought of, and if there's anything that's looked for in a clinic or in a community, is severe abdominal distension. And that's associated with ascites, or almost liver failure and fluid buildup within your abdomen, which is rare because the treatment programs are there. So you don't see this kind of extreme presentation, but you can get somebody with severe liver fibrosis, actually that looks completely normal. So the first thing we found is actually the clinical case definition is changing. You can't rely on that. We need to actually go out and do morbidity surveys rather than looking at somebody and thinking that they have a severe disease. The WHO at the moment relies on using current infection as a proxy indicator of morbidity or disease. They don't even bother measuring disease or setting guidelines for that. They'll have a cutoff for what they believe is the heavy intensity prevalence of schisto within a community, and that if you're below that or of some sort, there won't be any morbidity or any disease within those communities. I don't know where these guidelines came from. They're a bit arbitrary in my view, but we thought let's evaluate it. Let's evaluate it over and over and over in the cohort, in a meta-analysis. And we found that actually using current infection at all, regardless of a threshold you're trying to make up, doesn't make any sense. Schisto and its pathologies that it generates are progressive. They take quite a long time. So it makes more sense biologically that the current infection is not associated with your current morbidity. And this is something we've really been pushing for. And we even showed that actually, even due to potentially acquired immunity, differences in exposure patterns, if you measure somebody's water contact patterns in detail, even those will not be predictive of your current infection. Also, because current infection could be actually a long-standing infection. It could be a residual infection from imperfect treatment. So it's really, really a lot more complex than that. So it's time actually to think about revising those WHO guidelines and also measuring the disease a bit better. What we do find is, and that was quite shocking, is that we can see that there's interactions with HIV and hepatitis B with historical schistosomiasis that worsen that periportal or liver fibrosis. We also find that the increase in the likelihood of having this liver disease, it was thought that you wouldn't have this until your 40s or 50s. But actually, it's the change in the likelihood is happening as young as 25 years. And we have a really, really clear survivor bias within our population and also elsewhere, where the most severe cases, they're not going to live for very long. So it looks like they have a lower likelihood, but actually, sadly, they're dying. When we did a meta-analysis to kind of expand beyond the cohort and think a bit more on hepatitis B and different types of liver outcomes, we also were able to find that actually co-infection compared to even singular infection worsens a number of different types of fibrosis and cirrhosis, and even hepatocellular cancer, but that was for another species of schistosomiasis. So we were thinking, okay, well, it's not as simple as just tracking schistopathogenesis. The co-infections really, really matter. For improving also the diagnosis of fibrosis, we're working in areas where actually in SchistoTrack, we're pretty much employing all the sonographers across Uganda. There isn't really the capacity at the moment to go and kind of do these large-scale surveys, have the equipment. So we're thinking, how can we do this a bit better? Can we automate the diagnosis and the classification of the fibrosis? And Ellie within the group has been working on this for both actually images, which are curated, present some sort of bias from an expert, and videos, which could follow a protocol and you might use less expertise, which is a much harder problem. I've just shown a simple projection of the differences in the class prediction from the image model, but the video model performs equally well. And I actually showed right now probably what would be not the clearest cut model, because these fibrosis grades, even though they're presented as discrete or separate, they are continuous of some sort. And they do share similar views within the ultrasound. So we found out that we could train models that also learn the views that are being used, and that's what you would use to train a new sonographer. So we're hoping that these can result in a number of training tools. Then to reconceptualize what we think of as schistosomiasis morbidity, because schistosomiasis, again, is not a single disease. It's a complex set of interacting conditions. We've moved on to thinking about multimorbidity. What are all the interactions happening within the liver and the spleen? So Yin, a postdoc within the group, has been developing these beautifully sophisticated models where we're looking at 45 liver and spleen outcomes, understanding the dependencies between those outcomes and the dependencies between the models that we create for each of those outcomes to identify shared risk factors for overall hepatosplenic morbidity and groups of conditions as well. From that, we have some simple factors that matter that could be used for screening or triage that are current infection, again, doesn't show up to be important, but male, older age, fishing occupations, and living in Western Uganda, which we think is related to infrastructure because it has much poorer health infrastructure there. And then also have suggested, which is now going forward in a WHO AFRO advisory meeting later this month, but talking about getting clearer definitions as well of what it means to have severe schistosomiasis or danger signs, because a lot of countries will set goals of eliminating severe schistosomiasis, but what's that mean? And here we take the most severe outcome, which is gastroesophageal varices. This is essentially when you have such severe portal hypertension or high blood pressure originating from the liver that you get a backflow, and then you have enlarged varices within your esophagus or enlarged veins within your throat, to be very layman. And if those burst in these sorts of areas, if you had the right care and management, maybe you could get people blood supplies, but it's not the case here. So people do often die. They're dying from acute anemia from the blood loss, and they have these recurrent events. But we can find other characteristics that are associated with that, some of which, like hypersplenism, were not believed to be before. So in very enlarged spleen, we can also identify which patterns of liver fibrosis matter. The anemia, of course, is a clinical consequence. We can see redirection of blood flow through splenorenal shunts, and importantly, an enlarged main portal vein. And that's due to the fibrosis from schisto that's happening around the main portal vein. From when those eggs get swept back, you have that granulomatous response in the fibrosis. So this was actually so exciting. And we've had a huge impact from this. I've only been able to show really tiny little snippets from all the work that's going on within the group in the clinical work. We presented to the Minister of Health in Uganda. One of our study districts, the poorest one, that's by the DRC, has no hospital, has a dilapidated health center, doesn't even have permission to recruit the required expertise that they need to hire a sonographer. So we presented the burden of liver disease that we found, the few cases of mortality that we had already observed, the risk factors, and convinced her to publicly commit to building a hospital. They did. They started. So that is fantastic. That's still underway. I think we scared her a bit, but the goal was achieved. And we've started also working on training health workers on the difference between emergency and chronic care management with schisto, getting their input of what they think is difficult and not. Actually, as well, early on, some of this work had contributed to the Kigali Declaration. I was one of, if not the only, researcher invited to go to this meeting. This was right before actually the major COVID lockdowns in 2020, when we had plans for this work and lots of the earlier work that we had done. And this guides the NTD policy for all African states. And then, of course, we are revising how we should think about schistosomiasis morbidity. How should we diagnose it? How do we update protocols for getting the disease states when we have much better technology these days? The ultrasound protocol, which is the only thing available to diagnose any kind of disease related to any of the species of schistosomiasis, this is pretty much somebody looked at these images on ultrasound where they took a photo of them on some sort of old ultrasound and decided to draw out what they thought the patterns were. We can do better than that. So we've been working on that. But of course, this has been a great opportunity to kind of take a breath and think about what we've achieved so far. But, you know, I still have 30 or more years to go before Oxford's forced retirement age. So we have a lot more to do. The goal for the future is kind of thinking about schistosomiasis as a platform for those big global health questions, especially for new treatments for fibrosis, given its unique context. I want to work on creating one of the few African biobanks in partnership with the Uganda Ministry of Health, and integrating kind of really high throughput immunology. They have the platforms now in Uganda to do proteomics like Olink, so we're quite excited about that, so we could understand the mechanisms of fibrosis. We want to do individual-based modeling to mechanistically look at fibrosis progression. If you had these mathematical models that didn't have transmission as the outcome, but had morbidity progression as the outcome, then you could hypothetically look at kind of treatment scenarios. It's quite exciting. So a fellow in my group is starting that, Melissa. We want to scale up the cohort. This is something our new head of department, Prabhat, is interested in mortality studies. There's no good verbal autopsy tools for assessing schistosomiasis mortality. But this requires a lot more people than we work on. It's survey-based, but that's another goal of ours. To continue with the ultrasound imaging so that we can fully automate the diagnosis of liver fibrosis, train new sonographers, and provide risk prediction within a clinic. We're quite excited about that, and we're going forward with some of that work next year. But also really, really importantly, these days with funding, it's quite hard for students to get PhD scholarships. It's not the same. They're usually aggregated within doctoral training centers rather than individual scholarships. And it's even harder from my side to get PhD funding for African scholarships. So I am really set on trying to get more funding to support African PhDs in association with SchistoTrack to do their work and also earn a degree out of that. So I want to end with this. Another maybe media thing gone a bit interesting. I hope that I've made you a bit excited about Schisto and maybe inspired people who are yet to choose their career. If anyone ever watches this, that's quite young. So with the International Center for Mathematical Sciences and the Edinburgh Science Festival, again, they thought, hey, this person holding worms would be a great featured speaker again. They wanted someone who did applied maths in a weird context. So they hired a child's illustrator to animate my work. That was fine, that was great. Then, because it was a remote session, they showed this video to students in a Scottish primary school. And then they asked them to ask me a bunch of questions, and we had this kind of back and forth remote conversation. And one of them was about kind of what I do. And I was really advocating at that point that, you know, because I believe that actually, like, if you're going to go one way, maybe because this is how I come from a quantitative background and transition into the biology rather than the other way around. So I was saying, I think that every kid should go and study maths because you can get into anything you want. Then they had Lucas. He introduced himself. Meet Lucas, age 10. He does like maths. The interviewer, who's not me, who's coordinating the two things, is like, does this talk make you more or less likely to do maths as a job? Less, because this is weird. So I hope you don't feel the same way. And I want to thank, I didn't get a chance, there's so many awesome collaborators within this department outside that I've worked with. The funding that I've had has really supported this, but also my partner, Graham, who's very stylish, SchistoTrack shirt here. I haven't told him that I was wearing that. We have the youngest member of SchistoTrack coming in ourselves, so my life will be changing a bit in January. And then also my close friends who have been there, you know, day in, day out for all the funny little conversations or annoying ones. So thank you very, very much.